Okay, it's 8. Not many people showed up today, but that's okay. All right. You guys got the, before I start, you guys got the, were able to look at the gas exchange lecture I recorded? Okay, because you need to understand that to be able to understand what I'm going to talk about today. And I think I left it on this slide, the recording in which we have, I asked this question, and remember there were 2 recordings, one for blood pressure regulation and then the one that I started with gas exchange. And I ended up here asking you, we're looking at the effects of certain conditions like high or low partial pressure of CO2 and high or low pH. So let me review that really quick so that I can move to the next slide, to the one I ended in the recording. The main thing that determines, or the only thing really that determines hemoglobin oxygen saturation is going to be the partial pressure of oxygen. Normally you put this partial pressure of oxygen in arterial blood, is what is on this x-axis. Some people talk about partial pressure of oxygen in the air of the lungs, so alveolar PO2. But bottom line is that the partial pressure of oxygen, which correlates with the concentration of oxygen or the amount of oxygen in a certain medium, that being blood or air, is the main determinant of what the hemoglobin oxygen saturation is. Think about it. I gave you guys the example in the recording that if you have 10 students, and those 10 students are going to be the hemoglobin, so 10 hemoglobin, and you can carry 4 books, 1 book, 2 books, 3 books, 4 books, okay? Think of the books as oxygen, right? So each student can carry 4 books, and they have to take them from the classroom to the library, okay? If all 10 students have 4 books on them, then that is 100% saturation, okay? Every single student has all of the places they can carry books occupied. Same idea with hemoglobin. If I have 10 hemoglobins, which is way more than that, but if I have 10 hemoglobins, each hemoglobin can carry 4 oxygens and bound to those iron, right? And each hemoglobin is carrying it's saturated, is carrying the 4 oxygens, and I have those 10 hemoglobins all with 4 oxygens, that's going to be 100% saturation. And what determines that saturation is going to be the partial pressure of oxygen. If I have less oxygen available, then there's less likelihood of that hemoglobin binding the oxygen and carrying it. And that's what the job of hemoglobin is. The job of hemoglobin is to carry oxygen from the lungs, from the blood in those pulmonary capillaries, to the tissues, to the blood in those tissue capillaries. And then unload the oxygen so that the tissues can use it for metabolism. The hemoglobin has to let go of that oxygen. I'll talk about that in just a minute. But hemoglobin lets go of that oxygen. Now that oxygen is dissolved in the blood, so it's contributing to the partial pressure of oxygen in the blood, that PO2, and it can now diffuse into the cells, cross that cell membrane, down a partial pressure of oxygen gradient. Okay? Then hemoglobin picks up the CO2, right? It's also going to depend on the partial pressure of CO2, so that the tissues are extracting the oxygen and they're producing CO2 as a byproduct of metabolism. So at the level of the tissue, we're going to now generate venous blood. Venous blood has lower oxygen, high CO2, so that hemoglobin now in the venous blood is going to bind that CO2 and take it in that venous circulation back to the right side of the heart. Right side of the heart pumps it to the pulmonary circulation, where we are going to now unload the CO2. CO2 is going to move to that air in the alveoli of the lungs, and the oxygen in the air in the alveoli of the lungs is going to move into the blood. And we start the cycle again. So you see, hemoglobin is really just a carrier. Carries oxygen to the tissue, CO2 to the lungs. Okay? And what determines how much it can carry is how much oxygen is available for it to carry. So the partial pressure of oxygen is what determines how saturated it is. If now I have those 10 students and each of them is only carrying 2 books instead of 4, then I'm 50% saturated. Does that make sense? Oh, thank you. Let me put this one here. Okay. Does that make sense, guys? So if the MCAT, you get asked what determines hemoglobin oxygen saturation, what are you going to answer? Partial pressure of oxygen. Okay? The hemoglobin oxygen saturation is determined by the partial pressure of oxygen. Now, as I mentioned in the video, there are some conditions that are going to affect the affinity of hemoglobin for oxygen. The saturation is still going to depend on the oxygen, on the amount of oxygen, therefore the partial pressure of oxygen, right? But conditions as the amount of CO2, the amount of hydrogen ions or pH, and the temperature are going to affect that hemoglobin oxygen affinity. So how likely that hemoglobin is to bind that oxygen given a partial pressure of oxygen. So I talked about P50 with that, right? And I told you that P50 is the partial pressure of oxygen at which 50% of the hemoglobin is saturated. So what do I mean by that? Or how do I even look at that on a graph? Is I'm going to look at the 50%, which is going to be here, right? Remember, the y-axis is the saturation in percentage. So this is 50%. I extend that to the hemoglobin curve and then go down. And I apologize because my lines are not going to be straight. But this here is going to be between 27 and 30, let's say 27 millimeters of mercury. So the P50 for this particular graph is 27 millimeters of mercury, meaning that the partial pressure of oxygen, this is partial pressure of oxygen, at which 50% of the hemoglobin is saturated, is 27 millimeters of mercury. If that P50 increases, and I have a right shift of that hemoglobin oxygen dissociation curve, so this is a right shift, and it increases that P50, that means that that hemoglobin has a lower oxygen affinity. Why? Because now I'm going to need a larger PO2, a larger partial pressure of oxygen, to saturate that hemoglobin to 50%. And the opposite, if I now shift this curve to the left, so this is a left shift, which decreases the P50, this means that I have a higher O2 affinity. I need a lower partial pressure of oxygen to be able to saturate that hemoglobin to 50%. They use 50% just because it's an easy number to look at, right? And it's standard. All right? So what conditions can affect that hemoglobin oxygen affinity, can cause that right or left shift, is going to be things like pH. So here in this graph, I'm showing you the solid line hemoglobin curve and the dashed line hemoglobin curve. So the dashed line is a pH of 7.4, which is normal arterial blood pH. So your blood, if you don't have any pathology right now, it's 7.4. We're slightly alkalotic. And that pH, you're going to have this hemoglobin curve. If you were to decrease the pH, meaning your blood is now acidotic because you have more hydrogen ions. Remember, acidosis, or what pH does is it measures the concentration of hydrogen ions. So if you have more hydrogen ions, that means that the pH decreases, which means acidosis. Okay? So here we have a pH of 7.2. That means we have a lot more hydrogen ions. That means that the blood is now acidotic. That's going to cause a right shift. So that lower pH or acidosis causes a right shift, meaning that now we need a larger partial pressure of oxygen to saturate that hemoglobin to 50%. So the P50 increased. Okay? So we have a decreased hemoglobin oxygen affinity. Hemoglobin then is going to be more likely to unload the oxygen than to load it, okay? If we were to increase the pH, if we move now the pH to 7.8, that's going to cause a left shift. That alkalosis causes the pH, the curve to left shift. So you have a lower P50, meaning you need less oxygen to saturate that hemoglobin to 50%. Does that make sense? Everyone? Okay. Same with CO2. But CO2 has CO2 does two things. CO2 itself, if you have high concentration of CO2 or high partial pressure of CO2, that CO2 competes for the same binding site as oxygen. So if I have too much CO2, law of equilibrium, it's going to be more likely that that hemoglobin binds that CO2 rather than the oxygen, right? So high partial pressure of CO2 is going to cause a right shift, all right? Causes a right shift. It's going to move it this way, decreasing the hemoglobin-oxygen affinity. Okay, so that decreases O2 affinity. I'm going to just write it like that. If you have low partial pressure of CO2, then you're going to cause a left shift, which means that this will be a left shift, which means that we increase the oxygen affinity. Okay? So think about it. Well, no, before we think about it, let's look at the second way CO2 can affect hemoglobin oxygen affinity. So one is just having too much CO2 is going to compete for that binding site. It's going to decrease the affinity for oxygen of that hemoglobin. If we have too little CO2 and normal oxygen, then we're more likely to bind the oxygen than the CO2. That increases the hemoglobin oxygen affinity because they're not competing anymore. But the other way CO2 affects is, remember that equation I showed in the video. CO2 reacts with water very easily. And we always have an enzyme called carbonic anhydrase helping out this reaction or speeding up this reaction so that we form carbonic acid. H2CO3 is carbonic acid. And that dissociates into hydrogen ions and bicarbonate. And this equation can go in both directions, can go to the right or can go to the left. What determines if it goes to the right or the left? The amount of substrate, like in any chemical reaction. So if I have too much CO2, I'm going to have it be more likely to react with water and cause this equation to move to the right, producing more hydrogen ions. If I produce more hydrogen ions, then I'm going to produce acidosis, which causes a right shift. So not only having that high CO2 right shifts, but the CO2 reacting with water and making more hydrogen ions, making the blood acidotic, further increases that right shift, creates that Bohr effect. Does that make sense? And the opposite, if I have too little CO2, so if the CO2 is too low, right, this equation is going to move to the left to try to equilibrate that, right? So I'm going to have more hydrogen ions react with bicarbonate forming carbonic acid. That means that I have low concentration of hydrogen ions or alkalosis. And so having low CO2 not only left shifts because we have less competition for those oxygen binding sites, but also generating that alkalosis is going to further left shift and increase the hemoglobin oxygen affinity. Does that make sense? This equation right here, learn it because it comes in your MCAT all the time. It's acid-base balance. You probably saw it in chemistry or are going to see it in chemistry. If you take human physiology 2, you'll learn it again, and you're learning it now, and it will come in the MCAT for sure. Okay? They love this hemoglobin stuff in the MCAT. Okay, so just keep an eye on that. All right. Is this clear so far? This right and left shift. Okay. Now my question to you guys, and I'll ask it's a little bit more complicated what I'm going to give you, an extra thing to think about. I asked you at the end of that recording, I told you cells produce heat as a byproduct of metabolism, right? So remember, cells are going to extract that oxygen, use it to make ATP, but when they're doing that aerobic metabolism, they produce metabolic water, metabolic heat, and CO2, okay? So at the level of the tissues, that venous blood is going to have high CO2. It's going to be acidotic because we already know that too much CO2 reacts with water with the help of carbonic anhydrase that converts into carbonic acid. Carbonic acid dissociates into hydrogen ions and bicarbonate. So we increase the concentration of hydrogen ions in that venous blood. So venous blood is slightly acidotic. Not too much, but slightly acidotic. So it has high CO2, it's slightly acidotic because of the CO2. And tissues produce metabolic heat. So that venous blood is going to be slightly warmer. We learned that with the red mirabile. Remember the countercurrent heat exchange mechanism? So my question to you is, given these, would you expect hemoglobin to have a lower oxygen affinity at a higher or lower temperature? If you're looking at an adaptation, you want hemoglobin to load oxygen or unload oxygen at the level of the tissues? You want it to give the oxygen to the tissues or keep the oxygen? What do you guys think? Somebody? At the level of the tissues, we want to unload oxygen or load oxygen? What's that? Unload. Right? Is that what you said? You said load, okay. It's unload. At the level of the tissues, you want it to unload oxygen. Imagine this. Imagine hemoglobin is a bus, okay? And it has 4 seats. Those 4 seats for 1 oxygen. So it can carry 4 oxygens, right? Hemoglobin in us is going to be inside red blood cells as well. So at the level of the lungs, so imagine these are the lungs and these are the tissues, okay? And I need to bring those oxygen molecules from the lungs to the tissues to be used. It's like those oxygens are people and they need to get on that bus to go to work, okay? To be used in the work, okay? So I'm going to load those 4 oxygens, right? I have my bus full, 100% saturation. I close those doors. That oxygen is no longer available for partial pressure to contribute, I should say, to the partial pressure of oxygen, right? That oxygen is now sitting inside a bus. It's not going to be in the street contributing to the amount of oxygen I have available in that blood. Does that make sense? Which is the partial pressure of oxygen. Does that make sense? Okay. I close that bus. I go to the tissues. If I keep the doors of that bus shut, can those oxygens go out and do their work? No, they're loaded in the bus, and I'm not letting them go and do their work at the level of the tissues. They need to go to that mitochondria and help out to produce ATP. And I'm not letting them out of the bus. What do I need for that oxygen to be able to get from the blood in that tissue capillary into the cell? I need a pathway and I need a driving force. The pathway is there. Capillary is permeable to oxygen, the capillary wall. The cell membrane is permeable to oxygen. So the pathway is there. Permeability for oxygen is there. The driving force is the partial pressure of oxygen. But if that oxygen is trapped in the bus, trapped by hemoglobin, it will not contribute to that partial pressure of oxygen. Does that make sense? So what do I need to do? I need to unload that oxygen. I need to open my doors, let the oxygen out to that street, which will be the tissue, capillary blood, right? And now they contribute to a partial pressure of oxygen. They diffuse down their partial pressure into the cell, cross the capillary wall, cross the cell membrane, and then go into the mitochondria where it's used. Does that make sense? So I need to be able to load oxygen at the level of the lungs, fill up those buses, but at the level of the tissues, I need to let it go and load them. Okay? So I need to decrease the affinity of hemoglobin for oxygen so that hemoglobin lets them go. Okay? One thing that it's going to do that is that we're going to have at the level of the tissues less oxygen because the cells are continuously extracting that oxygen. Okay? So the partial pressure of oxygen in that venous blood now in the tissue capillaries, right? As the cells are extracting that oxygen, the oxygen is going to decrease. So if we have less oxygen, just look at the normal curve here. Less oxygen, I'm already less saturated. I'm 70% saturated. At the level of the lungs, I was 100% saturated because the partial pressure of oxygen at the level of the lungs was higher. But here, because the cells are extracting that oxygen and using it, then I have lower PO2, I have a lower saturation. So that's already helping me unload, that I have low partial pressure of oxygen at the level of the tissues. The other things that are going to help me unload is that I am producing CO2. Those cells are producing CO2, so CO2 is going to be higher. That's going to cause a right shift. CO2 reacts with water. We produce hydrogen ions that makes the blood slightly acidotic. That produces a right shift. So for the same PO2, 40 millimeters of mercury, I'm going to have a lower hemoglobin oxygen affinity because I am right shifting due to CO2 and pH. Now, do you think high or low temperature would help me unload or load hemoglobin? Do you think high or low temperature will decrease the hemoglobin oxygen affinity? Because that's what I need now. I need that hemoglobin to not want to grab that oxygen and let it go. So I need to decrease its affinity. I already did a little bit of that with low PO2, high CO2, and acidosis. At the level of the tissues, I'm producing metabolic heat. Do you think that metabolic heat is more likely then to decrease or increase the hemoglobin oxygen affinity? Decrease, right? High temperature is going to cause a right shift in that hemoglobin oxygen dissociation curve. So at the level of the tissues, I'm producing metabolic heat that's going to warm up the blood a little bit, and that causes a right shift, which further decreases the hemoglobin oxygen affinity. It's the perfect system, right? Because at the level of the lungs, I have high oxygen, high PO2, so I'm going to favor loading it, right? 100% PO2, I'm going to 100% saturate that hemoglobin. There's so much oxygen that I'm going to be very likely to bind that oxygen in all of the hemoglobin I have there. Then that hemoglobin inside the red blood cells go to the tissues, and at the level of the tissues, the cells are extracting oxygen, so the partial pressure of oxygen already decreased to 40 millimeters of mercury. That's going to decrease the hemoglobin oxygen saturation. Law of equilibrium. There's less oxygen there, less likely to bind to hemoglobin. Okay? And on top of that, I am right shifting that hemoglobin oxygen dissociation curve by increasing the amount of CO2, increasing the temperature, and decreasing the pH. Does that make sense? Yeah? Yes or no? I can explain again if need be. This is an important concept that you guys need to understand. So I need a solid yes, a solid no. Yes? Anybody no, and it's okay to say no? We're good? Can I move on? You sure? Okay. All right, the code is 112, 112, I think. Let me make sure. Yeah, 112. Okay, so how does oxygen get to tissues? As I mentioned already, oxygen needs to unbind from hemoglobin, right, to be able to diffuse into the cells. Because what is that driving force for diffusion? Is going to be the partial pressure of oxygen gradient, right? So let me get here. I'm trying to get my pen here to work. if I have the tissue capillary here, right? And this is the blood in the tissue capillaries. I'm going to label. Okay. Here I have my red blood cell with the hemoglobin inside, right? And here I have the cells in the tissue, okay? And the partial pressure of oxygen in those cells is low because the cells are using that oxygen to make ATP, right? But here in the blood in the tissue capillaries, this is blood, the partial pressure of oxygen is also going to be low if I remember very little oxygen travels dissolved in blood because it's not soluble, it's lipophilic, it's not soluble in plasma, which is 93% water, right? So in that blood, I only carry about 1% of the oxygen dissolved, meaning it's in solution and it contributes to a partial pressure of oxygen. So what I need is for hemoglobin to unbind that oxygen so that oxygen then can go here and make it so that partial pressure of oxygen in the tissue capillary blood, in that arterial blood that I bring to the tissue capillary is high. So now I have a partial pressure of oxygen gradient. High PO2 in that arterial blood in the tissue capillaries, low PO2 in the cells because they're consuming that oxygen. I have the pathway, I have the driving force, then oxygen is going to move to those cells. Does that make sense? So I need the hemoglobin to unload that oxygen, so now I can contribute to that partial pressure of oxygen, which is the driving force for oxygen to move into the cells, from the blood to the cells. So hemoglobin oxygen saturation is going to be low at the level of the tissues, as you see right here. This is the tissues at rest, a PO2 of 40 millimeters of mercury in that blood, right? And the saturation of oxygen there is about 70%. Whereas at the level of the lungs, where I have so much oxygen, because from the lungs, right, we have the lungs here. To put the lungs, this is the alveoli in the lungs. I don't know why it's not drawing. This is the alveoli of the lungs, right? And this is the blood in the pulmonary capillary, right? There's a lot of oxygen in that fresh air that you just inhaled, right? And you have now in those alveoli of the lungs. And the venous blood here has very little oxygen, right? So the partial pressure of oxygen in that venous blood is low. The partial pressure of oxygen in the air in the alveoli is high. You're going to have a driving force for oxygen to move into that blood in the pulmonary capillary and make arterial blood. Does that make sense? So at the level of the lungs, the partial pressure of oxygen in that blood is 100 millimeters of mercury, meaning that we're going to be able to fully saturate the hemoglobin to 100%. And if you measure, if you don't have any pathology and you use those oximeters I was talking about in the recording, with the little light beam, you should be between 98% and 100% saturated. You're at sea level too, so that helps. In 102, I teach about high altitude and how that affects all these. But at sea level, we should fully saturate if we're healthy, okay? But at the level of the tissues, because the PO2 is lower, then we saturate, that hemoglobin is less saturated, 70% in a healthy person. All right? Okay. Any questions about hemoglobin? And partial pressure of oxygen? And how we move that oxygen from lungs to tissues and tissues to lungs? We're good? Okay. Now I'm going to talk about what, and just one quick slide, what regulates the amount of oxygen, the amount of CO2, and the pH of your blood. Okay, so those three are physiological variables that you need to control and regulate. They are going to be homeostatically regulated with a negative feedback loop. You need to maintain the oxygen in your blood at within a certain range. The same with CO2 and the same with the concentration of hydrogen ions. Why? Because you need to have enough oxygen to satisfy the metabolic demands of the tissues, right? You need to be careful with CO2. Your tissues are going to be metabolizing, producing CO2, but if you have too much CO2, then you're going to become acidotic. Same idea, if you have too many hydrogen ions, you're going to become acidotic. And if the blood pH changes, right, then the intracellular pH, you're going to try to defend that and regulate for that. But if you cannot regulate it and bring it back to the homeostatic levels, then the intracellular pH changes. And if the pH inside the cells change, enzymes are not going to work properly, because remember, acidosis or alkalosis is going to change, denature those proteins, right? And the protein, if it gets denatured, if it changes its shape, it's not going to be able to perform its job. Function and structure go hand in hand. Does that make sense? If you have too little CO2, then also you're going to become alkalotic, or if you have too few hydrogen ions, you become alkalotic. That's a problem as well. And if you have, as I mentioned, too little oxygen, you don't satisfy the metabolic demands of tissues. If you have too much oxygen, oxygen is toxic. You don't want to have too much of that, okay? So it oxidizes, right? So it's going to damage tissues. So you have to regulate these physiological variables and keep them within a homeostatic range. So for us to be able to regulate, we need to have a negative feedback loop. And that's what we have here. We have these receptors that are going to sense the levels of these physiological variables, send that input signal or sensory signal to the central nervous system, where the information gets integrated. And if we need to do something about it, an output signal happens to an effector organ that is going to reverse the change in that physiological variable. There are two groups of chemoreceptors, central and peripheral chemoreceptors. So the central, I'm going to talk about the central chemoreceptors first. The central chemoreceptors monitor CO2 and pH. They do not, to this day, they haven't been able to find chemoreceptors in your brain that sense the levels of oxygen in the cerebral spinal fluid. But we do have central chemoreceptors that monitor CO2 and therefore pH. CO2 and pH go hand in hand. So these central chemoreceptors are in the brainstem, which is a part of your brain. It's the first part of your brain. So you have the spinal cord coming up, and right as it enters the skull, it becomes wider, and that's part of your brain, is the brainstem. And a lot of autonomic reflexes get regulated there. Cardiovascular, respiratory reflexes get regulated there. Vomit reflex there as well. I mean, hiccups, a bunch of them. So when you have here the capillaries, these are tissue capillaries, but these are capillaries in the brain, okay? Tissue capillaries in the brain. So you bring the blood, and let's say that blood has high partial pressure of CO2. So we have too much CO2 in that blood. If we have too much CO2 in the blood, we're going to have also too many hydrogen ions, because remember that CO2 reacts with water very easily, also in the blood, and produce hydrogen ions, okay? But these capillaries are very impermeable. They only let a few things cross, okay? So hydrogen ions cannot cross that capillary wall. CO2 can, because CO2 is small and it's lipophilic. So it can cross that phospholipid bilayer. So it crosses, and now you have too much CO2 in the cerebral spinal fluid, the CSF. Remember, the CSF is the fluid that bathes the neurons, okay? The neurons' environment. Remember, we talked about the glial cells keeping that CSF optimal? So that's the fluid that bathes the neurons, is the environment where these neurons are. So now if you have too much CO2, that's going to react with water. With the help of carbonic anhydrase, we produce carbonic acid, which dissociates into hydrogen ions and bicarbonate. So now you have too much, a high concentration of hydrogen ions. This CSF is now acidotic. You have high CO2 and you have high concentration of hydrogen ions. The pH of this CSF decreased, is reduced. And that gets sensed by the central chemoreceptors, which are going to send a signal right there in the same location, in the same brainstem, onto the respiratory control center. And that integrates that information. Because we have too much CO2 and acidosis, we're going to send a signal to an effector organ, so we're going to start breathing more. It sends a signal to our diaphragm, which helps us inhale more, and we're going to start breathing more. Larger volumes and faster breathing. So this increase in ventilation, it's going to decrease CO2 because you're going to breathe more, you're going to exhale more CO2. So that's going to normalize, so we go back here to this negative feedback, right? And we are going to reverse the change in that physiological variable. So this is a negative feedback loop. So that CO2 decreases again, and we start breathing normally again. Now, what happens with oxygen and with oxygen? Oxygen gets regulated by a different group of chemoreceptors called the peripheral arterial chemoreceptors. Let me write here they have the name of the two peripheral arterial chemoreceptors, so let me just write that down. Peripheral arterial chemoreceptors. So peripheral arterial chemoreceptors are going to be two types, carotid and aortic chemoreceptors. And they're located in the same location, in the same place as the baroreceptors. So the aortic peripheral arterial chemoreceptors are on the aortic arch, so where your aorta arches to descend. And the carotid chemoreceptors are going to be here. So you have the carotid arteries, which bring oxygenated blood to your brain, right here at the base of the skull. They bifurcate. Right in that bifurcation, you find the carotid chemoreceptors. So these chemoreceptors actually regulate the three physiological variables. They look at CO2, they look at oxygen, and they look at pH. So all three. So peripheral arterial chemoreceptors, which are carotid and aortic bodies, they are the receptors that monitor oxygen, CO2, and pH in your blood, okay, in your arterial blood. So if oxygen is low, if CO2 is high, or if the concentration of hydrogen ions is high, then that's going to send a signal through the same nerves as the baroreceptors, the glossopharyngeal and the vagus nerves, onto the respiratory control center. Okay, so these sensory neurons are going to be glossopharyngeal and vagus nerve, send a signal to the respiratory control center in the brainstem, and that increases ventilation. And if you increase ventilation, you're going to exhale more CO2, so you decrease the amount of CO2 in your blood, you're controlling for these. That will also lead to a decrease in hydrogen ions, so you bring back the pH to normal, and oxygen increases because you're inhaling more fresh air, you're bringing more oxygen to your body. Does that make sense? So you need to know these chemoreceptors. So central chemoreceptors only deal with CO2 and pH. Peripheral arterial chemoreceptors, which are going to be the carotid and aortic bodies, are going to regulate or sense or monitor both, all three, oxygen, CO2, and pH in your blood. Okay? And they are all going to send signals to the respiratory control center. So the central chemoreceptors through a receptor right there in the brainstem, the peripheral arterial chemoreceptors through the glossopharyngeal and the vagus nerve onto that respiratory control center in the brainstem, and they're going to cause a change in ventilation. Okay? So in this case, high CO2, high concentration of hydrogen ions, which is acidosis, and low oxygen are going to cause an increase in ventilation. The opposite will cause a decrease. So if I have too little CO2, too much oxygen, or alkalosis, then that's going to inhibit ventilation. Does that make sense? So expect one of these ones in the final. That's what I'm trying to get at, okay? like how it is regulated. You need to know these receptors. The same as the baroreceptors, you need to know these chemoreceptors, okay, both central and peripheral. All right, let me ask you a question here. Can ventilation change pH? What do you guys think? From everything you've learned so far, can I change the pH of our blood by changing ventilation? Think of the slide we just went through. What do you guys think? Who says yes? Who says no? Okay, good. Yes. A lot of no answers. You have to take a side, always take a side. So the answer is yes, you can. So actually, if you think about this, and before I move on here, think about this. What are you trying to do? You're trying to bring enough oxygen to your tissues, because that's what your tissues need to be able to metabolize and produce ATP. If we don't produce ATP, we die. Because our cells, that's the energy for our cells to do work. Okay? So the ultimate goal here is to make ATP. Sure, you can make ATP without oxygen, anaerobic metabolism, but that doesn't last very long. You become acidotic, you cannot sustain metabolism for a long period of time using anaerobic metabolism only. You need that oxygen. You need to be able to do aerobic metabolism. So ventilation and perfusion is what we learned with the cardiovascular system, circulating that blood. So ventilation from the respiratory system and perfusion in the cardiovascular system, their main goal, not only, but main goal is to bring oxygen to the tissues to satisfy their metabolic demand. Okay? And it needs to be perfectly matched. So if my metabolic rate is X, my perfusion in the cardiovascular system needs to be X, and my ventilation in the respiratory system needs to be X. I need to match those three. If I ventilate too much, right, and I don't need that much oxygen for metabolism, I'm wasting energy, because contracting those muscles to inhale is costly. Same idea, if I increase my cardiac output, but I don't need it because I'm bringing more oxygen than what needs to be getting to the tissues to satisfy their metabolic demands, then I'm going to also waste my energy. Contracting that heart is costly, takes ATP, right? Contracting muscle costs energy, right? So I need to perfectly match the three. I need to make sure that ventilation and perfusion are enough to bring what the tissues need. Otherwise, the tissues will extract what they need, and then I'm going to have a bunch of oxygen in that venous blood. No point there, right? Same idea, ventilation and perfusion need to be matched to each other. What's the point of breathing in too much and bringing too much oxygen if my cardiovascular system cannot take it to the tissues? It's useless. Same idea, if my cardiovascular system keeps perfusing, but I'm not ventilating enough, then I'm going to bring blood that is not fully saturated to the tissues. So those three need to go hand in hand, right? Metabolic demands of the tissues, how much oxygen they need to be matched to the oxygen supply. Ventilation and perfusion need to supply that oxygen for the tissues. So my tissues are going to be producing, or they're going to be performing at a certain metabolic rate, right? So if I have no change in metabolism, my tissues have not increased or decreased their metabolic rate. They're still consuming the same amount of oxygen. I'm, as a byproduct of metabolism, producing the same amount of CO2. But let's say that I increase ventilation. I'm going to breathe more. So if I breathe more, I'm going to bring more fresh air into my lungs. More of that oxygen diffuses into the blood. Remember, respiration and perfusion need to match each other. So then the cardiovascular system is going to pick up all that oxygen, increase perfusion to take that to the tissues. But bring the oxygen to the tissues. But at the same time, because you're increasing perfusion, you're bringing more CO2 to the lungs, and you're breathing more, you're exhaling more CO2. So you're producing, I'm just going to say here, just to kind of give you numbers I think is easier to visualize. And I'll let you guys go, guys, in a minute. Okay, so here, my metabolism is the same. Okay? So I'm producing 10 CO2s through my metabolism, right? Metabolic rate, tissues metabolize, they produce CO2 as a byproduct of metabolism, so I'm producing 10 CO2s. And normally, I would need to then move those 10 CO2s towards the lungs using my cardiovascular system, perfusion, and then I need to breathe enough to exhale those 10 CO2s, okay? But now I am breathing much more. I'm increasing my breathing, so I am exhaling 15 CO2s. That means that the amount of CO2 in my blood, that I always have some CO2 in the blood, is going to decrease, as you see right here. And if CO2 decreases, that's going to cause this equation to move towards the left. Because it's the law of equilibrium, right? If I have too little of this, then I'm going to try to compensate. So I'm going to try to make more of that CO2 that I'm having less of, because I'm exhaling more. So now more hydrogen ions are going to react with bicarbonate to form carbonic acid, and that dissociate into water and CO2 to try to compensate for this low CO2. So in doing that, I'm going to decrease the amount of hydrogen ions. If I decrease the amount of hydrogen ions, then I'm going to have a decrease in the concentration of those ions, which causes alkalosis, an increase in pH. So pH goes up higher than 7.4, so 7.6, 7.8, and I have alkalosis. If I decrease breathing, right, but my metabolism hasn't changed, so this one is the same, right? So I'm still, as a byproduct of metabolism, producing 10 CO2s, but I decrease breathing. So I'm only exhaling maybe 5 CO2s, and I'm accumulating CO2s in my blood. That increase in CO2, it's going to now drive that equation to the right, because I have too much of these. So it's going to be more likely to react with water, forming carbonic acid. Carbonic acid dissociates into hydrogen ions and bicarbonate. So now I have more hydrogen ions. A higher concentration of hydrogen ions means decrease in pH, means acidosis. Okay? So ventilation can be used to regulate pH disturbances. And actually, your respiratory system takes care of about 75% of pH disturbances because it's very quick. We can change ventilation really fast with the, like that regulation with the central and peripheral arterial chemoreceptors. It happens super quickly. But keep in mind, it can also cause it. So hypoventilation refers when you're breathing less than what you require for metabolism, and hyperventilation, hyper means more, you're breathing more than what you require for metabolism. And those two terms, you need to know them. Hypoventilation and hyperventilation. Okay, so hypo means you're breathing less than what you require for, it's comparing to metabolism. So hypoventilation, you're breathing less when you need for metabolism to satisfy those metabolic demands. Hyperventilation, you're breathing way more than what you need to satisfy the metabolic demands. So there's a mismatch between demand and supply. All right, I'm going to let you guys go. If you had trouble with midterm 2, I know we haven't returned, we return it in a week, by Thursday, but if you feel that you didn't perform well in midterm 2, please do talk to me right now, because you can put all your eggs in that last basket, which is the final exam, okay? So I want to make sure that if you're struggling with midterm 1 and 2, you reach out to us so that we can help you out with the final, okay? Don't leave it to the end. Talk to me now, next week. Hey, how are you?